Alright, so I did my project on determining the identity of KSC-DA, which is a bacterium that was isolated from the Ashwelot River. It was isolated by another undergraduate student last semester, Devin Aldebert, and my whole project was just determining what it was, because he isolated it, but we weren't quite sure on what the strain of it was. So I determined that it was Pentoia agglomerans, which is a species of gram-negative bacterium, and it is found in many different bioremediation projects, which is what this shows. Alright, so this figure shows the pipeline that I used, and those were all these steps were done through either Ron or Patrick, which are two different databases that we used. And through that, we found some reference strains that we used because we thought that it was Pentoia agglomerans. So we picked two different reference strains to compare it to, and those results are shown here. And these are how similar the reference strains were. I put them through both the databases to show the difference that they would have. And they're all pretty similar except for this second reference strain. And we also used Patrick to make a phylogeny chart, and it had the contigs from my project in here, which is next to a few other different Pentoia species, one strain that was very close to mine. This figure shows a bandage graph, which shows connections that usually you can't see between the contigs, and it allows you to visualize the de novo assembly. And so some future directions that you could take with this project would be to focus on a specific gene, or also to look at how similar the bacteria is to known pathogens. And these are my references.